little bit more to help you understand all of those muscle names that you have on your lab hit list. Sometimes if we can make sense of something or understand how something got its name, it tends to stick a little bit better. So you can use a table like this to understand or maybe get hints on how certain muscles got their name or make sense of their names. So some muscles have the action that the muscle performs right there in the name. Remember, muscle actions occur always when muscles are shortening, never pulling apart. So muscles always pull the bones, they never push the bones apart. So when we see an action in the name of a muscle, sometimes it's, we'll talk about some arm muscles today. Down here in the forearm, on the anterior side are all of your flexor muscles. Right? They'd be flexing the wrist or flexing the digit. On the posterior side are all of the extensor muscles, so the extension of the wrist or the extensions of the digits. And some of those muscles actually have that word right there in the name. So there's flexor carpi radialis and an extensor carpi radialis on either side of the forearm there. So flexor and extensor are part of the name. You have an adductor magnus down in your leg. You probably predict that's going to be adducting the thighs. So having the action in the name of the muscle sometimes is a great way to remember what that muscle does. Muscles can also have their location. So those two flexors I just mentioned, right, they're flexor carpi radialis. So flexor carpi radialis and extensor carpi radialis, I should say. They're both on the radius side of the forearm. So radialis gives us that indication, right, that they're on the thumb side, the lateral side of the forearm there. Sometimes the origin and the insertion are right there in the name of a muscle. So a great example of an origin and insertion muscle is this neck muscle. It's called the sternocleidomastoid. Its origin points are the sternum and the clavicles, and then it extends up to your mastoid process. So it's sternocleidomastoid is the name of the muscle. So right there, you've got O's and I's. Sometimes the orientation of the fibers, we'll talk about the different fiber orientations in a moment, but fibers can be arranged in a circular pattern. They call that an orbicularis muscle. So there's one around your legs, the orbicularis oris. Fibers can be straight. So rectus is the word for straight, like rectus abdominis muscles. The fibers or the fascicles are arranged in this really straight pattern. So sometimes that's right there in the muscle name. Shape and size. The trapezius muscle looks like a trapezoid. The deltoid muscle looks like a Greek letter delta, which is triangle shape. Pectoralis major is much larger than the pectoralis minor, just deep to it. Gluteus maximus is larger than gluteus medius. So the shape or the size is sometimes a part of the name. And then the number of heads or origins that a muscle might have. Biceps brachii has 2 heads. Triceps brachii has 3 heads. Biceps femoris has 2 heads. That's down in your thigh. So that's all part of naming some of these muscles. So if you can again make sense of how a muscle got its name, or relate the muscle's name to something, some characteristic about it, it might help with memorizing those O's, I's, and E's as well. The next slide describes different angles or directions that the fascicles can be arranged in. So remember that a fascicle is a bundle of cells surrounded by some perimysium, a little bit of dense irregular connective tissue. These little fascicles are like the little grain of the meat. Like if you're, meat eater, the little shredded meats, right? The little shreds, those are fascicles, bundles of cells. So fascicle direction is something that we can often see in our cats and in our human models even, right? They've drawn these little lines, or etched, I should say, etched these little lines on all of the muscles. That's meant to illustrate or demonstrate the direction that the fascicles are patterned in. So those patterns are described up here. A circular pattern is a common one for orifices. Muscles that surround an opening are often going to be circular in pattern. So we've got the orbicularis oris around our mouth, right, your lip muscles. The orbicularis oculi is around your eyes, like your eyelid muscles. And these circular muscles, when they contract, they close the orifice. They don't open and close, right? They just close. Remember, contraction, the action of a muscle can only happen when the muscle is contracting, not when it's relaxing. So we say it closes those orifices. Parallel muscles are muscles where the fascicles are running in kind of a parallel direction to one another, and they tend to look really strap-like, these muscles, kind of long and skinny usually. Parallel muscle arrangement, the fascicles typically run all the way from tendon to tendon. So origin tendon to insertion tendon, which means that the cells are that long, right? Because again, a fascicle is a bundle of cells. So that's what you expect to find in parallel fascicle direction. Convergent is described as kind of a fan-shaped fascicle direction. So typically kind of a wider origin and a narrow insertion is common on a convergent muscle. Pectoralis major is a great example of a convergent muscle with the wide origin point of the sternum and the clavicle, and then that pinpointed insertion over here on the proximal end of the humerus. So that gives us this convergence of the fascicles. And then pennate. Pennate arrangement is kind of interesting. The fascicles tend to run at an oblique angle to like the length of a muscle. So if we were to draw maybe a parallel muscle, right, with its tendon on either end, this would be the belly of the muscle. In a parallel muscle, the fascicles are all running, origin tendon to insertion tendon. But in a pennate muscle, the fascicles are going to be running at an angle, at an oblique angle. So no fascicle is going to stretch all the way from origin to insertion, right? Which means all these cells are running at an oblique angle. The pennate arrangement packs in many more cells than a parallel arrangement for muscles of the same size. And we would say that a pennate muscle would be much stronger than a parallel muscle, typically. So it generates much more force. This is a unipennate muscle with one kind of row of these obliquely angled fascicles. But there are examples in the body of bipennate. So one of your quadriceps muscles, it's called the rectus femoris. That guy's a bipennate muscle with fascicles arranged kind of like a feather, right, running opposite each other. And then there's multipennate muscles where you have more kind of bundles of fascicles arranged at that oblique angle. The deltoid muscle is an example of that. It's super strong for its size, especially. So generating lots of force is one of the characteristics of those pennate muscles. All right, on to muscle cells here. So myofibers or muscle cells are a little bit different in different muscle types. Turns out we have 3 different types of myofibers in all of our muscles, and every single muscle in your body is going to have all 3 of these. So it's not like you only find one type in the sternocleidomastoid and you only find another type in the erector spinae muscles. Right, we have a mixture, but some muscles in your body have like more of one of these than the other 2, for example. So predominant type of myofiber. So let's take a look at these myofiber types, these 3 types, and then we'll describe what kind of characteristics they have and where they might be more likely to be more abundant in a muscle. So the first fiber type is called the type 1 fiber. It's a slow oxidative fiber. These myofibers, they have lots of mitochondria. Mitochondria are the little organelles inside of the cell that are responsible for generating ATP, like a power plant. So they take one form of energy, like nutrients that we eat, and convert that energy. They break it down and convert it into ATP energy. And then you know that ATP is the only energy that the cells can use to do work. And there's a lot of ATP demands in our muscle cells. Right? Not just those sodium potassium pumps and the calcium pumps inside of those cells, but in all of these sarcomeres, we've got myosin heads that are pulling those actin thin filaments toward the M line. And every single cranking of the head of one head burns through 1 molecule of ATP. So that's a lot of ATP demand. So the more, the more mitochondria a cell has, the better, right? And muscle cells, all muscle cells have a ton of mitochondria. The cool thing about these slow oxidative fibers is that they also have this stored up glucose. So they also store glucose as glycogen. That's not in there actually, but that should be there on the slide. They have an ability to store this glycogen. So they have these glucose reserves, right, that they're able to keep inside of the cell. So that's great. They're not dependent on the glucose in your bloodstream, right? They have their own reserves. And then the thing that is listed up there is myoglobin. Now myoglobin is a protein that the cell makes. And it sounds a lot like another protein that I hope you've heard of before. You heard of a protein that sounds similar?
Hemoglobin?
Hemoglobin. Do you know what hemoglobin does?
It's like coagulates your blood.
It does what?
Well, like when you like bleed out, isn't that what like?
Oxygen transportation.
It's in your blood, and it's there to transport oxygen. That's its job. So hemoglobin transports oxygen. Well, myoglobin is even better at holding oxygen than hemoglobin is. It's almost like a magnet for oxygen. So what happens is blood travels through and around all your cells, all your tissues. Any cell that has myoglobin is going to have a real good ability to pull that oxygen out of the bloodstream. Right? It's excellent. So what this means for our slow oxidative myofibers is that they will never run out of oxygen. As long as you're breathing, you're putting oxygen in your bloodstream. And as long as there's oxygen in your bloodstream, that myoglobin is going to be the first to take it. So having myoglobin basically ensures the cells that they will never run out of oxygen. So they have exactly what they need to constantly make ATPs for the mitochondria to do their jobs. These slow oxidative fibers will basically never fatigue. They are able to contract all day, every day. We find an abundance of these slow oxidative fibers in your posture muscles. So think about the muscles that are holding up your head all day, right? Those posterior neck muscles, there's a muscle back there, it's not on your list, but it's called the splenius capitis, right? That's holding your head in this erect position. If that muscle were to ever fatigue or not be able to contract, your skull would, it's heavy, right? It would fall forward. Your posture muscles in your back, there's a group of muscles back there called the erector spinae muscles. We'll take a look at them. Those muscles have a huge abundance of these slow oxidative fibers. When you perform endurance activities, right? If you're like a marathon runner, then you are developing your slow oxidative fibers in your leg muscle. Anytime you're doing an activity for an extended period of time, it's those slow oxidative fibers that are going to respond the best and grow the best in response to that activity. So while they are everywhere in all muscles of our bodies, they are super abundant in those posture muscles. The fast oxidative fibers are the type 2As. So these guys have some glycogen, right? So that's that glucose storage protein that these guys have as well. So they have some glycogen. They have a little bit of myoglobin. So they're able to also draw oxygen, but maybe not quite as well as the slow oxidative fibers. These fast oxidative fibers, they're kind of like, maybe like the second best at endurance activities. They can go for a pretty good period of time. They can keep making ATPs, but because they have a little less myoglobin and because they have a little less glycogen, they're not going to be able to go forever, right? So these guys are going to be involved in more of your like endurance activities. They're not like super strong explosive myofibers, but they can go for a decent period of time as long as you have enough oxygen around. But if oxygen supplies are dwindling, then these guys might end up running out of ATPs due to the mitochondria not being able to really maximally make those ATPs effectively. So those are your fast oxidatives. These guys are really common in the leg muscles. We develop these all the time just by standing, walking. If you do any sort of leg exercises where you run or ride a bike or skateboard or anything, right? Most of that is going to involve leg muscle activities. So you're developing these fast oxidative fibers. The last one are the fast glycolytics, or the type 2Bs. So glycolytic fibers really don't have much myoglobin, which means they're just relying on the oxygen that's already there. But as soon as the mitochondria burn through that oxygen that's there, they don't have a way to supply their mitochondria with more oxygen. So what'll happen in the fast glycolytics is they're able to contract, but on the order of like seconds is kind of their maximal contraction. Under a minute typically is for the maximum contraction of these guys. These guys will run out of oxygen, which means they will run out of ATPs. Glycolysis, just the word that's listed here, anaerobic glycolysis, is a quick way to make ATPs without oxygen, without mitochondria, basically. But it's a really inefficient process. This is way beyond my class, but just a little like FYI for your information. When mitochondria have oxygen and glucose, from one molecule of glucose, they can generate something like 32 to 36 molecules of ATP, quite a bit. It's a really efficient process. Without oxygen, mitochondria aren't functioning, and the only way the cell can make ATPs from glucose, if they still have glucose, is through this process. It's called anaerobic glycolysis, and they just make two molecules of ATP from a single molecule of glucose. So it's really inefficient, and it will burn through the glucose really quickly. And then of course, the cell runs out of ATP. So just a little like, that's why these guys can go forever, and that's why these guys fatigue after less than a minute. Of course, when you rest, like let's say you're lifting weights and you're like maxing it out, you're using these fast glycolytic fibers for that, right? And then when you fatigue, you can't lift anymore, you have to rest, right? During that rest phase, the cell is replenishing its oxygen, right? And it's getting ready to go again. It's resupplying ATPs and getting ready to go again. So those are your fast glycolytics. Under the microscope, the slow oxidatives are the darkest ones. So these are myofibers cut in cross section. They're so dark because of that myoglobin. The fast oxidatives, those are going to be kind of like in between the two, and they still have myoglobin, so they're going to have some of this like protein staining inside, but no myoglobin really in these fast glycolytics. So they look really clear, really light staining under the microscope.
For like rock climbers and stuff like that who hang on for a very long time, would that be still fast glycolic muscle fibers, or? So
they've probably through training, right? You develop, all muscles have all three. They've probably developed more of these two within those same arm muscles.
Okay.
Yeah, it really depends on how you're training your body. If you're training your body by like lifting heavy weights and just going there, then you're training these guys right to the point of fatigue. But if you're building up to being able to like cling and hang, right, you're also training those other fibers.
The more endurance someone has, is it because they produce more ATP? Or does it just last longer?
So more endurance is directly related to the ability to produce ATP for a long period of time. ATP itself does not last any longer in somebody who runs and somebody who doesn't run. But the ability of the myofibers to make ATP, to basically to replace it. So there's this constant recycling that's happening. That's really what's preventing fatigue. Right? Fatigue is when you run out of ATP.
And does that process get started when you're training?
Yes.
Okay.
Yes. So the cells respond when you train. Like, I used to run, and so when I was running and training, I could run for hours, but I stopped, and then I started running again, And I was like, holy smokes, 1 mile is really like I'm winded and tired. But now I'm back up to maybe about 3 miles because as I'm running, my cells are getting super fatigued. And that first run where I'm running a mile and I can't make it, right? I'm exhausting all these cells. And then my cells respond by growing, by making more myoglobin, by making more myofibers, right? Maybe if I need more strength, whatever it is to respond to the activity I'm tasking them. So you run a mile for a couple weeks and then that mile becomes easy. And you're like, holy smokes, this really works because that's what's happening. The cells are responding to the exhaustion or to the demand by building the proteins they need to support the activities you're demanding of them. Yeah. And the same would happen if you go to the gym and you start lifting heavy weights, like 3 reps, and then you can't get it anymore. Well, you were strong enough, you are strong enough to lift that heavy weight because you did it the first time. But by the 3rd time, your fast glycolytic fibers have run out of ATPs. So you do that a few times at the gym, and a month later, you're able to lift that same weight 3 times, no problem. So you maybe move on to a heavier weight, fatigue them again, challenge those muscles, they respond by growing. Yeah, Charles, you have a question for Oh.
I wanted to say about working out that we say in the gym day 5 after heavy workout is the most precious one because of that compensation. So muscle recover with a little bit on the top of you needed before so you can lift heavier.
Yeah.
That's what you're talking about.
Day 5.
Mm-hmm. Day 5, yeah.
Interesting.
So you never want to miss the day 5.
There you go.
That's when you'll get the best
There you
result.
go. There's your training tip for the day.
I'm talking about large muscles.
Lifting.
Yeah, like legs and back.
Yeah. Awesome.
Is that day 5 from the first workout?
From the, yeah, day 5 of the recovery.
Oh, that makes
So working out every day, lifting weights is not wise. That's a bad thing, actually.
Your muscle, yeah, I mean, it makes sense. Your cells need that recovery time to be able to build, right? So by supplying your body with proteins, you're supplying with the amino acids they need to make myoglobin, to make actin thin filaments and myosin thin filaments and all of those things. Yeah, and that's all going to contribute to the bulk of the muscle.
So when you're like talking about running the mile and after running 1 mile you still like are fatigued, but then after we keep doing it like practice, you know, helps with your ATP. When you're sick, how does that process work? Because I mean when you're sick and you're like you feel fatigued, then that means your ATP energy is like running out, but then how do you get that back?
That's a great question, and it really, there's so much, like the word sick, right? There's so many definitions of sick. When you're running a fever, for example, at higher temperatures, all of your body cells are a little more active, and any activity in those body cells is going to diminish their ATPs at a higher rate than you're used to. So your metabolic rate is higher, is elevated, and that leads to this sensation of fatigue. Muscle aches that come with like fever and flu-like symptoms, that's all related to the higher activity levels for sustained periods of time that you're like not used to. So that's going to contribute to fatigue when you're sick with something like the flu and a fever. And everything else is, I would suggest, or I would say, is probably related to that, in that your body is spending energy and oxygen is going to the cells that are doing those jobs of healing, right? Trying to heal the damage that, because I mean, that's what sickness is. It's damage, right? Yeah.
When during this time, how does your ATP kind of like come back? Because like when you're talking about running, it would come back after like practicing running, but then this
ATP comes back, I mean, you could run a mile and feel super fatigued and out of breath, but you're fine again 5 or 10 minutes later. ATP comes back quickly, right? What the difference between running a mile on day 1 and running a mile on, day 5 or day 14 and feeling like you could keep going is your cells' ability to grow and respond, to build more myoglobin, to store more oxygen. It has nothing to do with with that immediate use of ATP, it's the long-term growth that allows the cell to then recycle ATP more efficiently or more effectively. Yeah, so that's what gives you that like longevity. They're all great questions. Cool. Okay, and then the last concept with our muscular system is that your muscles are actually perfectly positioned to serve as like a lever or lever that allows us to basically have pretty small muscles that are relatively efficient at lifting heavier loads. So just an example of a lever, like I, can only lift so much. I don't even know how much. I don't lift like that. But I can, pick up a big box like an Amazon delivery, and I can pick it up, and it's really heavy, and I can maybe bring it into my house and put it on the table or on a chair, right? Probably couldn't even lift it to the table. I'd have to lift it to the table. Okay, so let's say, I don't know, I'm gonna give myself some generous qualities here. Let's say I can lift like a 60-pound box. That's probably my max. And I'm gonna like waddle it in, right? If I put that same 60-pound box in a wheelbarrow and I lifted the wheelbarrow, that's no problem. It's the same 60-pound box, but by putting it in this lever system, right? This system where there's a pivot point, we call that a fulcrum. I'm actually making my load a little bit lighter, or I'm using my muscles a little more efficiently to carry that exact same load. That's what our muscles do by using our bones and these pivot points that we call fulcrums. So a fulcrum is going to be that fixed pivot point. The effort is always generated from the muscle contracting. And that effort, that contraction effort, is going to be less when I use a fulcrum than if I were to, try to lift this load directly with this muscle, right? So the load is the resistance or the force or the weight that you're trying to move. So that's the idea, that our muscles are perfectly positioned so that we don't have to have ginormous biceps brachii muscles to pick something up, right? We're using our elbows as the fulcrum point there. And it enables us to have these relatively small, efficient muscles. There's 3 types of levers, right? Like my wheelbarrow lever, that was an example. That's what we would call a second-class lever. It has to do with the order. So basically, like, where's the pivot point? Where's the fulcrum? The first one, if the fulcrum is in the middle, right, it's called a first-class lever. So you've got effort on one side, fulcrum in the middle, load on the other side. In this particular case, like scissors, for example, you're able to lift a relatively heavy load with relatively reduced effort. So here, the example in our body of a first-class lever are those posture muscles, like the splenius papillus back here. That's this guy, and it's the one that's really holding your pretty heavy head up, right? So the effort is the muscle. The fulcrum point is basically the articulation between like the occipital condyles and C1, and then the load is the weight of your head, right? It wants to go forward. So that's a first-class example. A second-class example where the fulcrum is at one end, the effort's at the other with the load in the middle. So that example here is the gastrocnemius muscle that you're looking at. This is down in the lower leg area. So in this case, the load is actually the body weight coming down through the tibia bone, right? That's in the middle. The muscle is back here, posterior to the tibia, and the pivot point is up on your toe, right? That's your fulcrum point. So again, that tiny little gastrocnemius muscle is able to lift, right, pretty much almost your entire body weight, just with a small amount of effort there. The second-class lever has the load in the middle, effort, and fulcrum on either end. Third-class class levers. So this is the majority of our muscles in our human body. They're going to be third class levers with fulcrum at an end, effort in the middle, load at the other end. So biceps brachii muscle, brachialis muscle, those would all be examples of third class levers. The fulcrum point is the joint that's moving, in this case the elbow, right? The load is the weight of the entire forearm. So they've described it out here in the hand, but it's really this whole forearm that you're lifting. And the muscle, the effort is in the middle, in the pull. So again, levers using these fulcrum points really makes our muscles pretty efficient and able to lift much heavier loads than they otherwise would if there was like a direct pulling on the load itself. So that's it. That wraps up our muscle organ system. No, that's not where I'm gonna go. Here. Before we get into the nervous system, I want to show you on Canvas a little animation that I've got posted there. So I know Tuesday's lecture was a big one, and there were a lot of like molecular anatomy in there. This, collapse these. There we go. So these animations here. Oh, just a reminder, the Unit 2 muscle homework packet, right? Definitely you should be able to complete this now because we're done with our muscle lectures. So don't put this off, make sure you get to it. But the next page here has these animations. So these I thought were really nice animations that kind of reiterated a little bit of what we talked about. I know when I draw things on the board, it's really static, and sometimes, you know, getting the flow of things is difficult to visualize. So these can help with that. Let's go for the events at the neuromuscular junction first. Not full screen, it's not too blurry. And sometimes the volume doesn't work on these. I'm not sure how to fix it, but we'll see if we can get the volume working.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal.
I'm going to pause it. Which membrane proteins are on the axon that are allowing that action potential to occur? Membrane proteins on the axon. We showed them at all the nodes of RANVA.
Voltage gated.
The voltage gated channels. Voltage gated sodium channels and voltage gated potassium channels. Okay, those are the ones on the axon. When they open, they let sodium flood in and they let potassium flood out. It's all facilitated diffusion. And remember, facilitated diffusion does not require any energy. So this action potential doesn't require any energy at all, right? None is used. It's just one set of channels triggering the next set of channels, triggering the next set of channels, right? right? Like a domino effect, all the way down. So if an action potential starts at the axon hillock, which is the beginning of an axon, then it will trigger those voltage gated channels to open all the way down until we get to the axon terminus. So that is the electrical impulse, that is the action potential. It's this sodium entering the cell, and the cell gets really, really positive, followed by potassium diffusing out of the cell, and the cell returns back to a negative state there. Right? Again, that wave of positivity followed by a wave of negativity flows all the way down the axon until we get to the axon terminus. Once we get to the axon terminus, which membrane proteins are found here? The voltage-gated calcium channels. So that's what's going to happen next. Right? When those voltage-gated calcium channels open, that's going to allow calcium to enter at the axon terminus. Okay, let's keep going. There's those vesicles.
Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels.
Cations are positively charged ions. So sodium.
Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative.
So the way I teach this is just to focus on the sodium. We don't care about the potassium leaving. Less relevant to the cell getting positive. So what makes the cell really positive? We're in the myofiber right now. It's the sodium diffusing in, right? So that's the part that I focus on.
Step 7. Once the membrane potential
I'm going to pause it right here and just show you, maybe if you're not paying attention, you'll miss it. This is the sarcolemma, right? This is the sarcolemma of the myofiber at the neuromuscular junction that had those acetylcholine receptors on it. Notice in this illustration, these invaginations of the sarcolemma. Those are T-tubules, right? What's next to the T-tubule? This web network material, that's the sarcoplasmic reticulum. So they've got it all here, and you can see just how abundant it is in the cell. There's a T-tubule here, there's a T-tubule here, there's another one behind it, there's another one over here, like they are all over the place. So my diagram on the board was super simplified, right, just to kind of keep it a little organized and clean. I think I have like a single T-tubule and a small little section of sarcoplasmic reticulum. But the reality is that stuff is like all over all these myofibrils. It's all over, all throughout the myofiber. So just to kind of wrap your head a little bit around that three-dimensional anatomy. Back it up just a moment here. Here we go.
Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma.
And the T-tubules.
Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
So it's the neuron up above and the myofiber down below still. So that was just the events at the neuromuscular junction in that animation. The next one on the same page is the actual contraction of the myofiber.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads and thick myofilaments form cross bridges with actin molecules and thin myofilaments. The formation of a cross bridge is initiated when calcium ions released from the sarcoplasmic reticulum bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position.
So I did not focus on how ATP hydrolysis steps take place during that whole cycling, but what they just showed was a myosin head that was bound, right? It had already power stroked. It then slumped and then recocked. Right? So that slumping happened when that new molecule of ATP came in, and then when it was broken down, that's when it recocked. You don't need to focus on the state of ATP, just the phases of the myosin head.
A cross-bridge cycle may be divided into 4 steps. Step 1, cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2, the power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step
We call that the M line, remember? The center of the sarcomere.
3, cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4, reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat.
Look at all that ATP being used.
And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
transcripts are posted here. I don't think they're captioned, unfortunately. But you do have a transcript of everything that that person says to follow along. Yeah, so hopefully that felt okay.
It really helped.
Reminder.
Thank you.
I get that whole process of working and everything, but is it all being, it's like all being controlled by you? Like your thought, like to move?
Yes. Yes.
So like you thinking to move is what initially like starts the whole process.
Your brain, it's the conscious part of your brain. It's called, well, we'll learn about it next week, but it's your primary motor cortex that initiates all of those movements. So, yeah, I mean, it gets to the point where you don't realize you're thinking about moving, like what I just did, but that was like part of my conscious brain and my speaking. I'm communicating something to you with my gestures.
But the signal comes from the certain area of vertebrae, right? Responsible for the area.
Yes, that's a great point. The direct signal to the myofiber comes from your motor neurons, and your motor neurons, cell bodies, are in your spinal cord. Yeah. So the axon will leave the spinal cord, but the cell body is somewhere in your spinal cord.
Is that how you're writing
Yeah, so we'll see. You guys are getting a little ahead of the game here. What is that all called? It's oh my gosh. Let's see. Let's see if that pulls up an image. This is a pretty simple image of it, but this is kind of the basics. Different parts of your brain are making conscious decisions, but eventually it's your motor cortex that's going to have a neuron that sends a signal down its axon. So when this guy fires, then the action potential travels down its axon to whatever level of the spinal cord the motor neuron that's controlling that muscle cell is located in. So if I'm moving my arm muscles, those motor neurons are going to be up here in like my lower cervical and upper thoracic region of my spinal cord. If I'm moving my leg muscles, then those motor neurons are going to be down here in the lower regions. So the motor signal comes from the brain neuron to that motor neuron, relays there. The motor neuron in the spinal cord then sends a signal through its axon out to the myofiber. That's what we drew on the board.
Okay, so that's why people get like paralyzed if they damage their spine.
Yes.
Okay.
Yes.
Like every time you bring your arm forward, that means that all the calcium is rushing in and like getting rid of that. And then every time you relax, that means they're being taken away. And that's just happening constantly in your body.
Yeah, that's exactly it. Calcium is constantly being released from the sarcoplasmic reticulum and then resequestered back into it when you're relaxing. And then you can release it again. So it's constantly recycling as well. Yeah, it's location, right? Where is that calcium? Yeah. Awesome. Well, let's take a break. We are definitely not going to do our full nervous system lecture today. But I do want to introduce you just to some of the bigger components, just understanding central versus peripheral nervous system. So I'll spend another like 15 or 20 minutes just introducing the nervous system today, and then we'll make up for it. We'll figure it out another time. We'll catch up. But take a good break right now. How about 10 minutes? And then when we come back, we'll just give this a little start.